Hello everyone, my name is Ujjwal Kamboj and today I will be presenting on macrolide resistance, and this is a key topic under antimicrobial resistance in agriculture, but it also has a major importance in human medicine. And we will explore what macrolides are, why they are matter, how bacteria resist them, and how we detect resistance and what strategies can help to manage it. So let's go further. So coming up to the macrolides, what are macrolides? So macrolides are natural antibiotic compounds that contain large macrocyclic lactone ring attached to the sugars. So the most common ones that are used in antibiotics are erythromycin, azithromycin, clarithromycin, and roxithromycin. So they are very versatile because some derivatives are used as antifungals like amphotericin B and nystatin, and some are used as antiparasites like levermectin, and some of are used like an immunomodulators like tacrolimus and sirolimus. So their mode of actions are different. So they bind reversibly to the 50S ribosomal subunit at the parasite and blocking the protein synthesis and stopping the bacterial growth. So coming up to the next slide. So why these macrolides are important? So they are effectively against the gram positive bacteria except enterococci, that's exception, and some gram negative bacteria. So crucially they work well against the atypical pathogens like mycoplasma pneumoniae and chlamydia trachomatis, legionella, bordetella, and pertussis and bordella, sorry. like in humans they are used for lower respiratory tract infections and community acquired pneumonia, certainly sexually transmitted infections and opportunistic infections like mycobacterium avium complex. And in animals, like mainly in they treat the bovine respiratory disease like that's called the shipping fever and swine pneumonia, and in poultry diseases like necrotic enteritis and canine infections like pseudoderma or Lyme disease. So they are valuable because they penetrate the tissue well and accumulate in the white blood cell and delivering the high concentrations at infection sites. So coming up to the next slide. So resistance mechanism. Bacteria resist macrolides through three main pathways. First one is target modification, second one is efflux pumps, and third one is enzymatic inactivations, and last one is rare. So coming up to the first one, the target modification. So it is caused by like methylation of 23rd like sRNA and at the adenine, and I hope you know they are the 4 base pair ATGC. So it just work at the adenine, and even though I just mentioned the A2058, and it mediated by the genes like A, B, C, and F. These are the main 4 genes, and this prevent the macrolides from binding, and it also causes the cross resistance to the lincosaminides and streptogrammin B, and this is called the MLS phenotype. So yeah, so second one, the influx pump. So these are like actively pump of macrolides out of the bacterial cells, and these are treated by the genes like MSRA, MSRD, and MEFA. MEFA, and these are like common examples. Coming up to the enzymatic activation, I told you before it is rare but it is reported. So example are MPHC, and these are encoded into the like phosphotransferase enzyme that inactivates the macrolides. So coming up to the next slide, like how did resistance evolve, like how they are evolved. That's the important and that's the interesting one. So resistance appears soon after the erythromycin was first introduced in 1950s, and it spreads in largely due to the horizontal gene transfer. There are like two, like there is a vertical gene transfer and the horizontal gene transfer. So in vertical gene transfer they are just come to the family, genus, species, subspecies, but in the horizontal gene transfer they just coming to the within the species, within the family. So this is transferred into the horizontal gene transfer via the plasmids and trans and sorry transport like transposons, and the examples are ARM-A, that is gene carried on like transopon like TN554 in MRSA, and ARM-C, that is plasmid borne in SSA. So heavy antibiotic use is in animals for the growth promotion or long-term feed treatment. They all accelerate the resistance. In today, resistance is widespread in both human pathogens, veterinary pathogens, and even the field pathogens like agriculture pathogens too. So coming up to the next slide, so detections of the resistance, like detections, how do we detect the resistance? So there are like some tests, like it is called a D test. So this is used for staphylococcus to check the inducible like clindamycin resistance, and it plays in the erythromycin and clindamycin disc close to this close together, and if the clindamycin like inhibit the zone that flatters near the erythromycin disc, so yeah, it is called as the positive D zone. Remember, it is called as the positive D zone. And so come to the M phenotype. So this is resistant to erythromycin but still sensitive to clindamycin and usually caused by the efflux pumps. And coming up to the PCR methods, like PCR testing like detects the specific resistant genes like MSR and MPH graphs, and yeah, and there is one more like MIC testing. This is especially for the veterinary pathogens like Mycoplasma bovis and Pasteurella multocrida and Manhamia hematolytica. So yeah, these are the common, like these ones that mentioned in the material. So coming up to the next slide is only the like therapeutic like strategies, like if the resistance is detective, so what we just gonna do. So if the resistance is detective, we can use the alternative antibiotics such as the tetracyclines, fluoroquinolones, or cephalosporins. And in veterinary practices, avoid the long-term feed macrolides, like these are common in pig and poultry production systems. Use the short-term targeted therapy instead of the growth promotion, and rotate the antibiotic classes to reduce the resistance selection, because if we just treat with the same antibiotic for a long term, there is more chances to grow the resistance. And in both humans and animal medicine, macrolides are preferred over critical human drugs like fluoroquinolones and extended spectrum, and yeah, to preserve the last line treatment. So coming up to the next slides, like these are the important notes, or these are the takeaway points. So the first one is resistant rates can be very high in some regions, like for example up to 90% of BRD pathogens in certain studies, and new genes being discovered like 48 in Staphylococcus xylosus, yeah. And efflux pumps are methylation mechanism usually confer the stronger resistance, but compared to the enzymatic activations. So there is a public health concern since resistance bacteria can transfer between animals and humans, like a zoonotic risk. So yeah, coming up to the summary. So macrolides are powerful antibiotics, but resistance is rising rapidly. So the main mechanisms that they target methylation, influx pumps, and enzymatic inactivation. And resistance, yeah, the resistance is evolved through the gene transfer. I told you it's a horizontal gene transfer and often spread by the plasmid and transposons. And the detection method we told about D-test, PCR assays, and MIC testing. And strategies, I just told you to careful the antibiotics use and use the rotate the drugs classes and restricting the misuse of the animal feeds. So the overall message, we need responsible antibiotic stewardships to preserve the macrolides effectiveness. And in last, I just want to thank you for your attention, and please feel free to ask any questions. And once I upload this video to YouTube, and yeah, let's work together to slow down the antimicrobial risks, and for the sake of the human and animal health. So that's all. Thank you.